welcome back to MS Biobrain. In our last video, we have learned some different topic that was about the pollination and the different agencies of pollination. Here, we are going to learn some different topic under the same chapter, reproduction in lower and the higher plants. My name is Manjushri, and here we are going to start a new topic, and the new topic is about the artificial hybridization as well as the double fertilization. So let's start. What is artificial hybridization? So it is basically the unnatural process that has to be done artificially. Double fertilization, as the name explains, it is the process of fertilization that will happen or takes place twice at a time. So how the process will be there and what are the different advantages or the significance of double fertilization, that all we are going to learn in this video. So don't miss any part of the video. Be here till the end so that you won't miss anything important. Okay, so let's start from the introduction of artificial hybridization. So basically, this process or this method is widely used. And why it is so? Because it is used for the crop improvement, and with the help of this process or method, we can make any desired variety of crops. And on the other hand, the double fertilization, it is a complex phenomenon in the group of angiosperms. Here in double fertilization, the two male gametes that enters into the ovule during the process of fertilization, that fertilizes two female cells, that is one egg cell and another one is the secondary nucleus. And the process of artificial hybridization takes place with the help of one particular process that is called emasculation. So what is emasculation? Emasculation is the process of removal of stamen or anther from bisexual, prevent the self-pollination. Bisexual flower means one in which both the gametes, both the sexes, the female and the male gamete will be there. In that we need to remove the stamen that avails for the artificial hybridization, as we can see in the diagram. When the flower is in bud condition and when it open, the emasculation has to be done by removing the anther or the male gametophyte. Okay, once the emasculated or the anther once removed, that is called as the emasculated anther, and after that the process of tagging and bagging will take place, as shown in the figure. That emasculated flower has to be packed in a polythene bag. That process is called as bagging, and we are placing the tag, just displaying that this particular flower has emasculated one, that is called as the tagging. Okay, so all this process, the emasculation, bagging and the tagging, that comes under the process of artificial hybridization. Now let's come to the double fertilization. So as I said, the double fertilization is the process in which the fertilization takes place twice at a time. Okay, now this double fertilization is a complex process. As said before, Notion in liliaceous plants discovered this double fertilization, and with the help of pollination, the pollen grains reach the surface of the stigma. This is the entire process that I'm talking. And there are some chemicals that helps to recognize the right type of pollen and allows its entry inside the ovule. Once the pollen enters inside the ovule, it start absorbing some nutrients and start germination. As the germination begins, there will arise a pollen tube. That pollen tube will enter and reaches into the female body. As we can see in the figure, the pollen tube is running throughout the style and reaching the female gamete. Inside that pollen tube, there are two male gametophyte. One out of those two male gametophyte, the one will fertilize with the egg cell and the another one will fertilize with the secondary nuclei. So this is why it is called as double fertilization, and these both cell will develop in later stages and will form the complete endosperm. Okay, now depending on the entry of pollen tube, it is divided into three different types. One is porogamy, in which the pollen tube will enters through the micropylar end, which is downwards. That is known as the porogamy, means instead of entering from the top, it will enters through the micropylar end or the microvalve, as shown in figure. The dark color pollen tube is entering through the micropylar end. This is porogamy. The internal process will be same, no change, but the only difference is entry of pollen tube. This is the one. Second one is the chalazogamy. Here in this process, the entry of pollen tube will be through the chalazal end, that is the topmost part of that female gametophyte. That is the chalazogamy. As shown in the figure, the micropylar end is closed and the pollen tube is entering from the chalazal end, and through one of the integument, the pollen tube will fuse and it will release the male gametophyte inside the nucellus or inside the female gamete or the female structure. This is the second one, chalazogamy. Now, the third one is mesogamy, in which the entry of pollen tube will be through one of the integuments, either the right one or the left one. But as we have seen in previous one, the chalazogamy, the pollen tube entering through the chalazal end. Here the pollen tube entering directly through the integuments, and those pollen tube fusing or entering through the integuments will release the male gametophyte or the pollen grains inside the nucleus for the process of fertilization. So are the three different types by which the pollen grain can make its entry into the female structure. Now let's see how is the process of double fertilization. So as I said, the double fertilization is the process in which both the male gamete will fertilize. So how will be the process begins? The pollen tube that absorbs some watery content of synergids and enters inside the ovary. It will fuse. It gets rupture and release the contents and the two male gametes. The siphonogamy, which is the process of carrying the two male gametes to the ovule, ensures the process of fertilization. In double fertilization, the fusion of both the male gamete takes place at two different places within the same structure, which is the main characteristic feature of angiosperm. So entirely we can say the double fertilization, in which both the male gamete will come inside the female structure and they will fertilize two different egg cells. One is egg cell and another one is the secondary nuclei. So in syngamy, a generative type of fertilization, there will be fusion of one male gamete with the egg cell to form a diploid zygote, which will result in the formation of an embryo. Triple fusion is a vegetative type of fertilization. The other male gamete fuse with the secondary nuclei to form a primary endosperm nucleus, PEN. In this process, both the male gamete participate and fuse at two different sites inside the ovary, and hence it is known as double fertilization. Now let's see what are the different significance of double fertilization. So this process utilizes both the male gametes and it is the unique characteristic feature of angiosperm. That is, the process is named as double fertilization. The embryo formed from the fusion of an egg cell and one of the male gametes will ultimately develop into a new plant, because that process follows the formation of embryo and the seed and the new plant. The triploid primary endosperm nucleus, PEN, that will develop and provides nourishment to the developing embryo, and the process of double fertilization that restores the diploid state. Then this process also prevents the polyembryony. That means the entire process is so useful. So this is it up. Thank you. For more details, you can visit our website, and if you like this video, hit the like button, share with your friends, and don't forget to subscribe our channel so that you will be getting the new knowledgeable videos on the different topics under the subject biology. So this is it. Thank you so much. Bye bye. Take care.